Adaptive immune response. Now you are having the knowledge on innate immunity. So it is about the first line defense. So now we are moving to adaptive immune response. So usually first response to particular antigen called primary response may take week or more to develop. Immune system remembers pathogens on subsequent exposure, term secondary response. It is related to adaptive response. Adaptive immunity, like innate immunity, we divide into humoral immunity, which contain antibody production, and cellular immunity, which contain cytotoxic T C cell mediated and T helper cell mediated activities. So usually in humoral immunity, antibody mediated eliminate extracellular pathogens, while cellular immunity leads to intracellular pathogen elimination. So overview of the humoral immunity, that is mainly driven by B lymphocytes, later termed in plasma cells. B lymphocytes synthesize or develops in bone marrow. B cells may trigger to proliferate into plasma cells. That's how it goes. Plasma cell produce antibodies. Antibodies produced when antigen binds to B cell receptor. Some B cells produce memory cells. So this is the overview again. So you can see this antigen present in cell, which present to T helper cell, at cell as to cytotoxic T cell. We'll go further detailing later on. And which proliferate and differentiate of activated T helper cells, naive to activated T helper cells, TH cell, and cytotoxic toxic T cells with the presence of antigen present in cells. It be bound in those T cell receptors. It proliferate and differentiate into activated cytotoxic T cells. Both are activated with induced apoptosis. It's like NK cell, granzyme mediated, perforin mediators, and trigger apoptosis, fast ligands like TH cell or T helper cells. One way activates macrophage to activate a macrophage, whereas it provides the signal to B cell. And with the same time, B cell is activated with the presence of extracellular antigens. Now it proliferates and differentiates into activated B cells. For that, there is a co-ligand signal is required. Then it turned into plasma cell. Plasma cell initially produce IgM antibodies, but class switching, they produce IgG and other antibodies, mainly IgG. That's how it goes. So about the overview, cellular immunity, it's mediated by T cells, T lymphocytes, matured in thymus, divided into two subsets, cytotoxic, denoted by TC, or helper T cells, other TH. T cell receptors help to antigen recognition, what I said earlier. Bit of lymphoid system, I think you know about the introduction of immunology we have discussed. So it is contain lymphatic vessels, secondary and primary lymphatic organs. Secondary lymphatic organs, you know, lymph nodes, spleen, tonsil, adenoids, mold, appendix even. Organism situated strategically allows for initiation of immune response from nearly any place in the body. Secondary lymphoid organism. Primary, you know, bone marrow and thymus. Antigens. We learned about bit of antigens, so we call it antibody generator. Then it's term antigen. Why merging these two words? Initial part. Antigen that cause immune response term immunogens. Protein and polysaccharide induce string response. Lipid and nucleic acids often do not. It's less. Recognition of antigen directed at antigenic determinants of site is called epitope. It's an epitope, and in antibody site is paratope. So counterpart is antibody. It's basically Y-shaped structure. So two arms, two sides antigen binding is possible. Let's term this part variable region is a constant region. Usually, so two chains are heavy. So these are two chains, heavy chains. Two light chains, bounds with hydrogen bonds. Each heavy and light chain has constant region. So it's a constant region up to here. heavy and light chain has variable region unique to each antibody. So this particular is unique for this particular antibody. Imagine this is dengue IgM antibody, dengue type 1. So it's specific. So they can bind on this epitope. This area is called paratope. It can bind only dengue 1 specific site only. This region binds to a specific antigen and it's known as FAB region. FAB region. So it's the place. Further, you can see disulfide bonds there. Multiple disulfide bonds are there. Heavy chain, light chain, variable region, constant region. So antibody antigen binding is very much important in immune response. One way neutralization, because antigens are maybe virus or bacteria or their cell wall constituents, their particular organelle constituents, so that they can evoke immune response leads to sepsis. So once the presence of antibodies against specific, they can go and catch them and prevent further cellular activation. So it's termed neutralization. And immobilization and prevention of adherence further. With the presence of these Y-shaped antibodies, they neutralize and also adhere and precipitation. Leads to agglutination and precipitation. Clumping of bacterial cells by specific antibody. Bacteria more easily phagocytes. It leads to opsonization. That's what it's happening. Once it's bound, particular organism, it's more easy to phagocyte. So also it leads to complement activation. Classical pathway leads to activation. Also then antibody dependent cellular cytotoxin. So it directly activates NK cells with the presence of this FAB, FC portion which bound to NK cells and trigger apoptosis and release of NK cell mediated damage. That's how it goes. So those are the functions of antibodies. So what are the types of antibodies? 5 classes. IgM. It's a common one. First antibody to respond to infection. 5 to 15%. Structure, it's a pentamer. 5 Ys are there. Found on surface of B lymphocyte as a monomer. When in B lymphocyte, B cell receptor like it's as a monomer, not as a pentamer. In the circulation, it's a pentamer. Only antibody that can be formed by the fetus. More efficient than Ig in the complement activation. So those IgM and IgG both cause complement activation. This is the one which are much more strong, IgM. IgG is the abundant one. Structure is a monomer. That's the only antibody which can cross placenta. IgA is a mucosal immunity found in secretions. Structure monomer in serum, dimer in secretions. Breast milk, mucus, tears, and saliva. So in the GI tract, respiratory tract, even urethral tract, genitoureteral tract, urethra, IgA provide good protection against invading pathogens. IgD is a minimal amount, less than 1%. Structure monomer, maturation of antibody response itself. Ig is barely detectable in circulation in healthy individuals. Structure monomer activates in allergic reaction. Always Ig is associated with type 1 hypersensitivity. So about bit of the clonal expansion or selection of lymphocytes. When antigen introduced into body, only appropriate antibody bounds. For that particular B lymphocyte activation is important. For that, initiate multiplication of specific antigen, process flow, clonal selection. Repeated cycles of cell division generate population of copied antibodies. Term clonal expansion. Without sustainable mutation, stimulation, cells undergo apoptosis. So that what is happening is, stem cell is there, they produce precursor cells and B cells, naive B cells there. So activated B cells with the presence of specific antigens. They are now antibodies containing activated B cells. With the repeated cycles of cell exposure, there is a repeated proliferation, leads to plasma cell formation and go for memory cell production. lymphocyte characteristic include initial they are immature. Antigen specific receptors are not initially formed, naive. Antigen receptors have but not encountered antigens. Activated, able to proliferate, have bound antigen. That's how it goes. They are effectors, descendants of activated lymphocytes. So it's termed effector cells, able to produce specific cytokines. Plasma cells, T helper and cytotoxic T cells, they are effector cells there. There are memory lymphocytes as well. Remember antigen on subsequent exposure. They produce vast drastical antibody IgG and minute IgM against those pre-programmed or pre-exposed, pre-sensitized antigens. They form antibodies. Antibody response, antigens bind to B cell receptor. So antigen once bind causes B cell response. In many cases, B cell needs confirmation from the helper T cells remember in that particular image. Antigen enters the body and is phagocytes and processed by macrophages. It's mainly neutrophil also there. These macrophages destroy antigens into the fragments, present a portion on the surface of the macrophage next to self-antigen. Macrophages are called then antigen present in cells. Process antigen combined with specific T helper cells with the appropriate receptor. Antigen present in cell releases substances to activate TH cells. TH cell activates B cells to divide into and differentiate, produce plasma cells and memory cells. That's how it goes. So here antigen. So once B cell, it's engulf antigen presentation. B cell also antigen present in cell. Degrade internalize antigen into peptide fragments with the presence of T cells and co-stimulated signal. They tend to B cell present peptide fragment into groove of the MHC class 2 molecules on B cells surface. Now, if the T cell receptor of a TH cells bind to one of these fragments, then cytokines are delivered to the B cells, initiating the process of clonal expansion. That's its interregulator. When these antigen fragments, they are presented with MHC class 2 to be T receptors, T cells. So once these are activated, they produce cytokines to paracrine fashion and activates further B cells and goes for the clonal expansion, leads to production of IgM and further IgG antibodies. So usually I said in the primary response, this is primary response, not a innate immunity, first line defense. Okay, this is a primary response in adaptive immune response related to humoral immunity. So always lag period of 10 to 12 days occur before antibody detection in blood. So once antigen or invading pathogen is there, it takes 10 to 12 days to sensitize for that particular reactions to occur and leads to clonal expansion. That's how it goes. Once the clonal expansion leads, it's initially form IgM. That's why it appears early, right? With the time, it goes IgG activation also there, but still it's in a primary response. That's why in acute infection, antibody or serology is less value because it takes few days. If we detect molecules from here in using PCR, nucleic acid amplification test, it's easy to detect. It takes, even antigen detection will be helpful, but antibody detection, we need another reference value as well. Initial value, otherwise we don't know when it was exposed. Imagine it has a high value, we don't know because it takes months, usually 3 months. Like, so imagine in this stage, we don't know it's due to this antigen or another antigen exposed. That's why we want to do one sample and do another conversion sample. But when to confirm the diagnosis, it takes 10 to 12 days, maybe 14 days. So in that particular period, if you are not treated, patient will die. It's related to mainly to antibiotics, so bacterial infection. So that's the less use. So about the primary response, there's a lag period. So activated B cell proliferate and differentiate into increasing number of plasma cells as long as antigen is present. So it goes for secondary response. Net results is slow steady increase antibody titer, still in the primary response. But in the secondary response, when now memory cells are also formed, when this particular antigen specifically exposed here, now within the short lag time, there's IgM production and massive IgG production, it's the secondary response. So this is very much important in dengue diagnosis. We'll discuss. Antibody response. Over time, some B cells undergo changes. Therefore, with the repeated exposure, it terms affinity maturation, class switch in formation of memory cells. So once those antigen is repeatedly exposing and leads to antibody affinity maturation. Affinity maturation means binding ability of particular antigen, its term affinity. So once with the time passes with the repeated exposure, the affinity will be high compared to naive young antibodies or initial phase of infection. Class switching. So that is production of IgM, later switch into with the presence of interleukin 4 and 5, which goes to produce IgG, formation of memory cell. It's another advantage. So class switching, I said what commonly. So this is the overall picture what I mentioned earlier, discuss. So here again we will discuss APC antigen presents antigen to T helper cell. Antigen present in cell with the MHC class 2. The antigen is there. They present to T cell receptor. Same time, this is a co-stimulatory signal. B7 ligand, CD28 receptor. T helper cells there. So now with the presence of these two co-stimulatory signals and presentation, it goes to activated state for T helper cell. So these T helper cell, now activated T helper cells interact with B cells via CD40 ligand. Activated T helper cells interact with CD40 ligand with B cells. At the same time, MHC binding is so there. That's initiate proliferation, differentiation, and it leads to formation of one-way plasma cells, activated plasma cells which produce IgM, and this is class switching, leads to with the presence of interleukin 4, which goes for Ig and IgG antibody formation, and activated plasma cells with the presence of interleukin 5. Again, class switching IgA, at same time, they goes for memory cells. So usually TH cells secrete cytokines that determine class switching. So this is the activated T helper cells which secretes. These are interactions. You see antigen presentation, maybe this is a macrophage, T helper cells. Now this is, this is T helper cells. Usually they not directly act with antigens with the presence of antigen present in cells. So like a cascade. T helper cells, is activated, they then in turn activates and give the signals and acts on B cells and leads to proliferation and differentiation and go for the memory cell formation. So antibody response, memory formation, it's usual Ig antibodies term memory. Produce IgG antibody. So memory cells next exposure, they mainly produce IgG antibodies. So that's why it's term memory like. So this is the secondary response. So memory cells responsible for swift effective reactions of secondary response. Vaccine exploits phenomena of immunological memory. So once vaccine is there, it's a weakened antigen-like. So once exposed, now memory cells are formed with the presence of secondary expose. So now it's going to drastically immune response producing IgG protective antibodies and protect the human from that particular invading pathogen. There are other things like T-independent antigens can stimulate antibody response, leads to activated B cells without helper T cells. Few antigens are T cell independent. Like some polysaccharides and lipopolysaccharides act as T independent and T independent antigens. B cell receptor bite antigens simultaneously leads to B cell activation. B cell receptors those polysaccharides without T pathway, they directly activate B cell. That's why it's termed T independent. Earlier it was here T dependent. Here directly activates, but it's rare. Few exceptions. General characteristic of T cells, further bit, have multiple copies of T cell receptors. Receptors have variable site of antigen bounding. Role of T cell differentiate from B cells. T cells never produce antibodies, mind you. T cells arms with effectors that interact directly with antigens. T cell receptor does not react with free antigens. That's the other thing. Antigen must be presented by antigen presenting cell, what I said earlier. So general characteristic, there are two types of MHC, major histocompatibility complex molecules. During antigen presentation, antigen cradle in groove of major MHC molecule or major histocompatibility complex. So two types, MHC1 binds endogenous antigens, endogenous, bind exogenous antigen, class 2. So you can appreciate here, there are different binding sites are there, like constant and variable, not a Y-shaped one, sperm alpha and beta chain. So here, professional APCs, dendritic cells and macrophage, and B cells. There are key features. BC, dendritic cells and macrophages, phagocyte express receptor for apoptosis for damage associated molecular pattern and pathogen associated molecular pattern. It's important in sepsis, localized to tissues, localized to cells, zones of lymph nodes following activation. Likewise, there are express stimulatory molecules following activation. But usually atypical antigen present in cells are mast cell, basophil, eosinophils, and ILC3 cells. Inducible expression of MHC class 2 molecules, they are there. So if you know this part, that will be enough. For the comparison I put here for the, but you know what are the antigen present in cells. These are professionals. Further, macrophage present to T cell via MHC antigen. Dendritic cells present to T cell via MHC antigen. B cell present to T cell via antigen captured by surface antibody, because B cell receptor is always antibody, and that's IgM monomer and MHC antigen. Activated T cell present to T cells via MHC antigen. That's how it goes. T lymphocytes, antigen recognition and response. General characteristic, two major function T cell populations, cytotoxic T cells, proliferate and differentiate to destroy infected or cancerous cells. That's how T cytotoxic activity is going on. They are having CD8 marker while they are possessing MHC class 1 molecule on their surface. T helper cells, they are having MHC class 2 and they are termed CD4 marker. Crystal of differentiation. Okay, CD and CD8 and CD4. Stimulates other T cells, postulates immune response. That's how T helper cells. Here it directly acts, but there's a helper to T cell. Stimulation is there. That's how it goes. For these mature helper cells, antigen presentation, T cell receptor, CD4, it's MHC2, CD4, MHC2. Here it is antigen presentation, immature CD8 cells, T cell receptor via MHC class 1 leads to mature cytotoxic T cells, activated cytotoxic T cells, activated helper T cells. Antigen recognition and response. Function of cytotoxic cell, induce apoptosis in cells infected with viruses or intracellular microbes. Same as NK cells, they trigger apoptosis. Destroy cancerous cells as well. So cancer or tumor immunity, this is very much important like NK cells. Nucleated cell degrade portion of proteins load peptide into groove of MHC class 1. They are MHC class 2, here 1. MHC class 1 molecules recognized by circulating cytotoxic T cells. Cell destroyed by lethal effector function of T cells. T cell release preformed cytotoxin to destroy cells. So they identify MHC class 1. Like in NK cells, when all the other cells, every cells contain MHC class 1. When viral is, that particular cell is infected, NK cell looks, if it is MHC 1 flag is there, they don't act. But in the infected cell, MHC class, that particular flag is deflagged or removed. So now NK cell acts here. This nucleated cell degrade portion of the proteins load peptide in the group of the MHC class 1 molecule. They go with the MHC 1. MHC class 1 molecules recognized by circulating T cytotoxic cells. And they act. That's how cytotoxic T cells act. NK cells different way. About helper T cells, I said orchestrates immune response, recognize antigen presented by MHC class 2 molecules. MHC class 2 molecules found only in antigen presenting cells. If T helper cells recognize antigen, cytokines are delivered. Cytokines activate antigen presenting cells to destroy antigen. That's how it goes. There are no direct cytotoxic effect. These are helper cells. Other thing is role of T helper cells. We discussed earlier for the B cell activation. We have discussed in several places. If T helper cells encounters B cell bearing peptide, MHC class 2 complex producing its stimulate production of cytokines. B cell is activated in response to cytokine stimulation. B cell proliferate undergo class switching, affinity maturation, and memory cell formation. So other than that, role of TH cells in macrophage activation, what we discussed earlier. Macrophage routinely engulf invading microbes resistant to lysosomal killing. If TH cells recognize macrophage with engulf microbes resistant to killing, TH cells activate macrophage by delivering cytokines that induce more potent destructive mechanisms. That's what's happening. Engulf material, macrophage degrade proteins, and they present to MHC class 2. It's antigen presenting cell. Then it's activated. T cell is the co-extremely target delivery of cytokine activates macrophage. Secretion of the cytokines with this binding, and in turn it activates macrophage into activated macrophage and destroy these engulfed peptide fragments or antigens. That's how it goes. Natural killer cells. We have discussed about this. They don't have an antigen receptors. Recognize antigen by means of Fc portion of IgG antibodies. Allow NK cells to attach to antibody coated cells. Action augment adaptive immune response. It's a bridging like. Natural killer cells recognize destroy hot cells via no MHC, what I said earlier. That's it, it goes. So this is the pictorial presentation overall. Secondary lymphoid organ, primary lymphoid organ, formation of immature T cells, B cells, with the presence of viruses, antigen, APC antigen presenting cells, how naive cytotoxic activated, naive helper cells activated. So then we term into with the presence of co-stimulated signal, it goes for plasma cell formation and antibody production. At the same time, T helper cells with activates macrophages as well as cytotoxic T cells, and they go and act intracellular pathogens. Thank you.